So we've talked about using this linkage to be able to create maps, chromosome maps of different organisms. And we have so many different types of mutants, particularly in Drosophila, maize, which is corn, and mice. We have extensive chromosome mapping in these organisms. We have many different known phenotypes that correlate with different genotypes. And also, ethically, we can cross any of these that we would like to do. It's no problem. We want to cross somebody who's recessive in this trait with somebody who's dominant in that trait, and we want to see if it's on the same chromosome. That's no problem to set up crosses like that, and many of them happen very, very quickly. However, we can't do the same sort of thing with humans. Humans in the past, we were able to do some mapping of it, and that was based primarily on pedigree analysis. So going back through, looking at families, seeing how different traits were inherited. And what they did is what they call the log score method, or log of the odds favoring linkage. And what they did was just assess probability that a particular trait were linked or not linked. And they did this by looking at many, many different pedigrees over many, many different families, and just seeing how different things were being inherited together or not being inherited together. So you can see here what we know of the Drosophila genome. And you can see many of these genes are mapped. Many of these are going to be very physical traits that we can see the phenotypes of very, very clearly. Okay, these are going to be things like, tan body, miniature wings, curly wings, these sorts of things. Vestigial wings, brown eyes, rough eyes. And when we're talking about many of these different phenotypes, if you're looking at them under the microscope, the Drosophila eye actually provides us a great way of, it's very large, and many of these genes are going to affect what it looks like and how well it functions. Okay, many of the others are going to be things like bristles, the little bristles on them, their wings, their abdomen, things like that are going to just be very visible things to notice. Okay. So we've moved past that, obviously, now with the dawn of genetic sequencing. And so what we do for chromosome mapping today is we use very expensive machines, but the cost of an individual genotyping or sequencing has gone down dramatically. What they do is they take DNA markers. They're going to be short segments of DNA. We know the sequence, we know the location of them, and we consider them landmarks, or like little signposts. The restriction fragment length polymorphisms, or RFLPs, and microsatellites are going to be polymorphic sites. Many of the people are going to have variations within these sites. And there's millions of different possible combinations and variations that you can get. So by looking at these, it helps to give geneticists the ability to identify and locate related genes.